Okay, good morning everyone, good morning. Good morning Sadhguru, good morning Saima. Good morning Jayashree, good morning Shafia. Good morning Priya. Okay, good morning all. Okay, so friends, if you quickly recall, in the yesterday class we have started the cancer biology topic. Good morning everyone, and this is the slide what we were discussing about. Okay, is it? What did I tell you friends in this particular slide? If you all people quickly recall, we understand that the cancer growth is depends on the defective control of cell death or the cell differentiation. In the yesterday class we have seen one question, right? The differences between the cancer cell and the cancer stem cell. See, I want every one of you to remember one of the significant difference. Good morning, good morning. That is friends, cancer stem cell can always undergo the process of self-renewal, but whereas friends, cancer cell never ever undergo the process of self-renewal. I want you people to remember this particular point without fail friends. Okay. Now, so in the yesterday class, we have studied some of the characteristics of the cancer. See dear, self-renewal you can understanding na Kaur. Self-renewal means it is giving rise to itself dear. But what do you mean by the division dear? Division, listen. When I use a word division, two things will happen. In the division means, listen, it can give rise to the daughter cell also, and it can give rise to the itself also in the uncontrollable manner. Whenever it is giving rise to itself in an uncontrollable manner, it is basically give rise to what? It is basically give rise to the cancer. But here, when the self-renewal stem cell giving rise to itself and it is giving rise to the daughter cell, and the daughter cell do not have the ability or capability to undergo the uncontrollable division. It will undergo the normal division. Hence, there is a no formation of the cancer. The differences between the self-renewal and cell division is, during the process of self-renewal, it can give rise to only itself, only one copy. Keep it in your mind. It will only give rise to itself only one copy. But during the cell division, it can give rise to the multiple copies of itself. Whenever it is giving rise to the multiple copies of itself in an uncontrollable manner, undoubtedly it gives rise to cancer. You're getting? But in case of self-renewal, it can give rise to itself only one copy. You got the point? Good morning all, good morning. Okay, so friends, now listen. What is metastasis? At the same time, what is the angiogenesis? Friends, understanding these two steps are very important. Please pay attention. Okay, listen to me for 20 minutes continuously, friends. Okay, listen. Metastasis. See friends, one point is very clear. What I have told you about the metastasis, the tumor cells are going into the other healthy areas and it starts developing the new colonies. That is what I told you about the metastasis. Now the point I want you people to remember here, when a particular cell wants to enter into the other area, first it has to show the process of what friends? Invasion. What do you mean by the word invasion? Moving into the adjacent area. And friends, when I use a word, it is moving into the adjacent area. Continue. How it will move to the adjacent area? Undoubtedly friends, via blood vessels or lymph vessels to the another part of the body. So friends, this particular process only we will be calling it as the angiogenesis. Kindly don't get confused with this particular statement. Anyway, look into the diagram. Everyone pay attention towards the diagram. Understand it very clearly. Friends, you can see here, initially the tumor is present at the connective tissue. Initially, or imagine the tumor is present at the epithelium. Okay, tumor is present at the epithelium. Friends, everyone listen. This tumor from the epithelial cell, it is basically entering into the liver actually. Friends, all of you try to understand what exactly I am saying. How can it move from epithelial cells to the liver cell? How is that possible? Every student pay attention, friends. First, we have discussed very clearly. Tumor cell speciality is what? Even though they are having the boundaries, what did I tell you, friends? It will break the boundaries. So what you have to understand, now this particular tumor cell break through the basal lamina. Okay, it got break through the basal lamina. Now it has to reach us to the adjacent other tissues such as the liver. So now how they have to reach? See, all the students look into the diagram. So friends, they are traveling through the group of blood vessels, or they are traveling through the lymphatic vessel. Now the point is very clear. From where this blood vessels has come from? These blood vessels are of normal cells. So what I want you to know here, the tumor cells uses the blood vessel and lymphatic vessels of other cells. ORR, I will show you the animation. ORR, they can make their own blood vessels. Kindly don't get confused with that point. They can make their own blood vessels. So friends, that particular process only we can call it as angiogenesis. Now everyone has to listen my statements very carefully. An example. 1,000 cells are moving. 1,000 cells are moving in the blood vessel or lymphatic vessel, friends. You have to believe out of 1,000 cells, or hardly 1 to 2 cells will survive. What's the reason? The reason may be multiple factors. You can take environmental factors, you can take nutrition, you can take availability of the oxygen. That's why our older people, even scientists, always suggest that doing fasting once in a week extremely good for the health. It is extremely healthy. Especially friends, whenever we are under the fasting, the angiogenesis process will be extremely inactive. We know very well in this particular lymphatic or blood vessels, friends, the cancer cells require abundant amount of oxygen. They require abundant amount of oxygen and they require more nutrients whenever they are traveling through the lymphatic and blood vessels. But the point is what here? So during the fasting, where is the nutrients in our body? We did not take the food only, and oxygen level also extremely low in our cells. As a result, friends, most of the tumor cells which are moving through the lymphatic and blood vessels, they will die because of the lack of nutrients and lack of oxygen. And this is the same thing was discovered by a Japanese scientist, which he awarded a Nobel Prize in the year 2019 also. Anyway, see friends, I told you that out of 1,000 cells, hardly 1 cell or 2 cells, 1 or 2 cells will survive. Friends, all the students listen to me very carefully. Whatever the 1 or 2 cells are survived, every student listen, this is the blood vessel, right? It got escaped from the blood vessel and entered into the new tissues. So that process we generally called it as what, friends? We generally called it as the extravasation. What do you call it as, friends? Extravasation. Then it entered into this particular damaged area. Sorry, it entered into the new area. What is that new area? Liver. Friends, listen to me. How many cells are entered into the liver? 1 cell or maximum of 2 cells, that's all. But friends, even though 1 or 2 cells got entered, but I want you to understand, friends, it has proliferate uninterruptedly and it has formed a group of cells in the liver. Originally the tumor was developed in the epithelium, and from the epithelium it break that particular layer, traveled through the blood vessel and lymphatic vessels and escape from that particular blood vessels, entered into the liver and developed a new colonies completely. That's a simplified point. And One more important step you have to remember, friends. You know, if we treat, if we diagnose this type tumor before metastasis, we can cure it very fast. We can cure it very fast. Either we can use the both combination of both radiation and chemotherapy. We can cure it faster. But friends, nowadays we are having very advanced techniques are there. Even though it entered into the newer areas, can be cured. But you know, friends, we can cure. There is no doubt about it. I am not denying that we can't cure. We can cure. But you know, you cannot give the guarantee that the tumor can be reappeared. Because friends, you have to listen to me. During the process of metastasis, it is not restricted to one area, right? During the process of metastasis, it is moving to many areas. So you can't remove all the organelles wherever the tumor was formed. That's impossible, which leads to the death of death of a person. So what we have to do before the process of metastasis, if you diagnose the cancer in a correct way, we can treat it with several combination therapies and we can prevent this particular metastasis very easily. Don't forget that. Okay. So friends, till this particular slide, whatever we have studied is the characteristics of a cancer. Now we have to understand what are the various causes of the cancer. Friends, everyone listen to me very carefully. If you quickly recall, while defining the cancer, what is the word I have used? I have used a word that cancer is a genetic disease. Correct na? Cancer is a genetic disease, but what did I tell you? It cannot be inherited. Why I have used the word it cannot be inherited? The reason behind that, I am calling it as a genetic disease because it is caused by the changes. Changes in what? Because it is caused by the changes in the gene sequences. It is caused by the alterations in the genes. It is caused by the mutations in the gene level. That's why I call it as the cancer, but it is not getting inherited all the time. So all the students listen to me carefully. Cancer is caused by the changes. What do we call that particular changes? That changes only we can call the mutations to the DNA within the cell. Kindly do not get confused with that particular point. And we all people know very well what is the speciality of a DNA. We know very well DNA is made up of many genes, is it? DNA is made up of many genes which is packed inside the cell. DNA is the one which will, I told you right, it's like a hard disk which can store the entire information. It will only tell how the cell has to grow, how the cell has to divide, how many number of times cell has to divide, when the cell has to be differentiated, what kind of function has to be performed by the cell. Everything is guided exclusively by this particular DNA molecule only. We all know very well about it. Of course, true. Now friends, listen. Suppose example, every student listen. DNA is not guiding properly. What do you mean by that? The gene which is present in the DNA is not guiding the cell to divide properly. The gene present in the DNA is not guiding the cell not to differentiate, not to perform its function. When the cell loses its ability to divide, when the cell loses its ability to differentiate, when the cell loses its ability to perform its function, undoubtedly what is happening, friends? It becomes cancerous. Not only that, friends, you know, nowadays 20 to 25% of the cancers are occurring because of the environment. Environment also plays one of the crucial role in causing the abnormalities in the gene level which leads to the cancer. And sun and other type of radiation, friends, most of the people will think that bacteria are, see, I am giving you one line, okay, you should not get confused with that particular line when they ask you in the exam. Bacteria and viruses causes cancer. You know, this statement is a very contradictory statement. It always makes us confused. You have to understand, every bacteria and every virus cannot cause the cancer. That is not a true statement. Certain bacteria and certain viruses, they are having a carcinogenic activity, which means they are having the ability or capability to cause the tumor formation. Only that. So if they give the question, if they give the statement all the bacteria and viruses cause the cancer, that is a wrong statement. Every bacteria cannot cause the cancer. Every virus cannot cause the cancer. HIV virus cannot cause the cancer. But you know, right? I will explain you. HIV virus destruct the immune system. As a result, it will help other virus to enter. What is that other virus? Example, some other herpes virus, which can cause the cancer because it is having the carcinogenic property. So you don't get confused, friends. All the bacteria and all the viruses cannot cause the cancer. The bacteria and virus which is having the carcinogenic property, they will only cause the cancer. Kindly don't get confused with that point. Next, radiation. And friends, till today it was not proved. What not proved? Whatever the energy or whatever the radiation that is coming from the cell phone have not been found to cause the cancer in the people. So what I mean to say that, friends, there will be a no documental evidence. There will be a no documental evidence. There will be a no proof. There will be a no documental evidence. There will be a no proof to prove that the cancer can occur even from the radiation of the cell phone. There is a no proof. But we all people commonly know that X-rays, gamma rays and another many radiations, CT as well as majorly mRNA, MRI and PET scans, they are all causes severe radiation. They may cause the cancer even in the fetus level also. That's why in the hospitals they will keep the caution boards. Pregnant women are not allowed to enter into the X-rays because, friends, X-rays can easily penetrate into the fetus level and it can cause the genetic abnormalities or gene changes in the baby also. But what I would like to say, please don't get confused. I would like to say chest X-rays, CT, MRI. I am not denying that. I am not denying that they not cause the tumor. They will cause the tumor. But what I would like to conclude, my friends, the risk of cancer from these medical procedures are very small. And the benefit from having them is almost always greater than the risk. Now you can ask me a question, sir, then why you are calling the radiations will cause the cancer? That is true when you go for the multiple times. When you go for the multiple times. Friends, example I am telling you, if you take one mRNA for the brain, one MRI for the brain, it's not idealized. It's not idealized not to take the mRNA for the period of 1.5 to 2 years until and unless you are having serious complications. It's not required. When you, some people, they will undergo the MRI twice in a 6 months period. Twice in 6 months period. That is very dangerous. Because you know, right, when we are taking the mRNA, we have to go to the mission and we will expose our head to the radiation up to 10 to 14 minutes. It's not a short duration. It's a very long duration period. So that's why we should not go this MRIs, CT scans very periodically until it is mandatory or until it is required. When we expose it to 1 or 2 times in a in a long span, that won't cause any kind of damages. Kindly don't get confused with that particular point. Okay, now friends, listen, this is what radiation, and the most important one I want you to remember is chemical carcinogen and biological agents, from where we are getting the questions in the exam. Chemical carcinogen. See friends, many kinds of alkylating agents, every student keep it in your mind. Many kind of alkylating agents are cancerous in nature. Okay, and at the same time, asbestos, nickel, cadmium, radon, vinyl chloride, benzidine and benzene, they are also having the carcinogenic property. But what is the point? They are having the carcinogenic activity at very low concentration when they are working alone. But in any condition, asbestos and vinyl chloride got combined, then their carcinogenic property will be increased more and more. I want you people to remember, friends, any chemical carcinogen, what it will do? It will do only two things to cause the cancer, friends. All the students listen. Opportunistic cancers, correct. So everyone listen. This particular carcinogens will do two things. What is that? They will cause the cancer by altering cellular metabolism. This is one way of showing the tumor, and another way of showing the tumor is damaging DNA directly in the cells. These are all the two ways how a chemical carcinogen or chemical agent can act like a carcinogen. Most of the time, friends, they will cause the damage to the DNA. They will cause change in the base sequence. We all people commonly know that adenine has to pair with thymine and guanine has to pair with cytosine. Because of treating with the chemical carcinogen, we have read in the genetics chapter also, we have read in the molecular biology chapter also. Good morning, Karishma, continue. We read in those two chapters in terms of a mutation. So what is happening here? Adenine will go and pair with what, friends? Cytosine. As a result, what happened? Entire base sequence got changed. Correct na? Which leads to a great change in the sequence, obviously, which causes the mutation, leads to the cancer. And friends, the most important one which we have got twice, 4 marker question. Twice we got the 4 marks. Very easy, very straightforward. And what you have to do is you have to remember it. Here you don't need to use your brain. You have to remember it mostly. Okay. Biological agents, I have already given you an example. I have given you a statement that all the bacteria and all the viruses never ever cause the cancer at all. So listen to me. 6 infectious agents have been shown to be carcinogenic primarily by including chronic inflammation. I mean to say, listen my friends, listen to me very carefully. Originally that infectious agents generally can show the chronic inflammation. In addition, they are also having the carcinogenic properties. Examples, 2 major viruses, HBV and HCV, and bacterium infections like Helicobacter pylori. Helicobacter pylori basically causes what, my friends? Ulcers. We know, right? If you don't treat the ulcers properly on a right time, there is a great chance that it can easily convert into the cancer. Kindly don't get confused, friends. So two type of viruses having the carcinogenic activity, 1 bacteria having carcinogenic activity, and 3 worms, Scytoscoma hematobium, Apstoris viverrini, and Chloronyx sinensis. See, anyway, HBV, HCV, Helicobacter pylori is not difficult to remember. What you people has to remember, which is difficult, that is the 3 worms. I want you people to remember these 6 examples without fail. See friends, these biological agents can cause many type of a cancer. Even they can cause the breast cancer, they can cause the endometrial cancers, they can cause the prostate cancers. Okay. And friends, actually these biological agents will cause great imbalance in the hormones, even which leads to the abnormalities in the cell growth and the reproduction, which leads to the various types of the cancers. So what I want you to remember is this table. See, this table is present in every reference book. It is present in the QB also. It is present in the CARP also. It is present in many books. Lordish also it is present. Direct question. They have asked twice from this particular slide only. And you people to no need to remember the area of incidence. What I want you people to remember is, especially friends, the family. The virus or the bacteria belongs to which family, and tumors or cancers associated with it. These things I want you to remember. It's very easy to remember. You know, no friends, viruses generally having RNA as their genetic material. But we also know very well, viruses, all the viruses not only having RNA as their genetic material, some of the viruses having DNA also has their genetic material. Kindly do not get confused with that particular point. So listen to me very carefully, friends. DNA viruses, everyone listen. There are 3 viruses are there who is having DNA as their genetic material. Everyone pay attention and remember, here you have to remember papilloma virus, hepatitis B virus, most important, which is asked in the exam, Epstein-Barr virus. If you can remember, friends, I am requesting all of you remember the name of the family also. Otherwise, remember the virus and what kind of tumors it can be formed. Papilloma virus. Friends, don't get confused. This is very confucius, friends. Remember the statements very carefully. Listen to me carefully. Papilloma virus, friends, it contains many strains. Many strains are present. But if you ask me a question, sir, generally, I can say that the generally papilloma virus cannot cause the cancer. What it can cause is the warts, which are the benign only. You might observe, friends, especially on the chest, on the stomach region, even on the back. See, certain warts will appear. The wart will be like this. Exactly will be like this. You know why that wart occur? Just listen to me carefully. Suppose example, there is a small skin cut occur. Through the cut, virus enter over there and it can form that particular wart-like structure. So when this wart appeared all of a sudden because of a small infection, people will think that it is a cancerous. It is not a cancerous. It is just a wart which is caused by the viral infection, that's all. That is only we can call, friends, benign. See, I am using a word generally papilloma viruses causes warts. But friends, there are many strains of the papilloma virus are there which is having the ability or capability to cause the cancer. They can cause the cancer of uterine as well as they can cause the cancer of cervix also, cervical cancer. Hepatitis B, one of the very dangerous, which can cause the liver cancer. Friends, if there is a cancer affected the liver, there is only one way of a treatment, that is the liver transplantation. If there is a no liver transplantation, liver never work properly, obviously it leads to the death of the person. But there is a no doubt, friends, nowadays whatever the liver transplantations are occurring throughout the world, the transplantations are occurring very successfully. There is a no doubt about it. But you know, friends, even the transplantation occurs very successfully, the liver is not working as before. The liver is not, the new liver which got transplanted, it is not working very healthy as before. So as a result, even though after successful transplantation also, people are not surviving for not more than 2 to 3 years. It depends. So that's why liver is one of the very prominent organ that we should take care, because many medicines, you know, right, you just imagine, liver got damaged, we will take the medicine to control that. And whatever the drug or a medicine we have to take, we will take, that has to be metabolized by what? Liver only. If that medicine is not metabolized, that excess product accumulated in the liver will damage it further. That's why liver is one of the prominent organ that has to be taken care. Now, Epstein-Barr virus, please remember, don't get confused. The cancer of the B lymphocyte, we will see in the immunology also, which we widely call it as, friends, Burkitt's lymphoma. I want you to remember these 3, very easy to remember. Epstein-Barr virus, Burkitt's lymphoma, hepatitis B virus, liver cancer, papilloma virus, generally causes the warts, and in certain cases it causes the uterine and cervical cancers also. Now whatever the three I have explained it to you, these three are the viruses that can cause the cancer who is having DNA as their genetic material. I have also told you the viruses are having RNA also as their genetic material. So please remember, which are belongs to the family of retrovirus. I want you to remember compulsory. Human T cell leukemia virus, then friends HIV, human immunodeficiency virus. See, now you listen to me. Actually, in the exam they have asked is Epstein-Barr virus, HTLV and HIV they have asked. And friends, listen to me. HTLV, human T-cell leukemia virus, it will cause majorly lymphoma, the cancer of the T cells. What do you mean by the word cancer of the T cell? We all people know, right, that T cell actually differentiated into T helper cell and cytotoxic T cell. Friends, we all people know very well, if cytotoxic T cell wants to be active, what do they require? We require T helper cell. I will tell you in the immunology very elaboratively. At the same time, if a B cell wants to be activated, what do we require? Compulsory we require T helper cell. I mean to say, T helper cell required for the development of the cytotoxic T cell, T helper cell required for the development of the B cell also. If the T helper cell got mutated, friends, there will be a no cytotoxic cell, there will be a no development of the B cell, there is a no production of antibody only. Very dangerous cancer it is. And next one, human immunodeficiency virus. Friends, please kindly don't get confused with this. HIV virus never caused the cancer. Never ever. You all people commonly know that. HIV virus can act on which cells, friends? We all people know very well. HIV virus act on the T helper cell. And you also know very well, anyway I'll explain in the immunology also, more specifically HIV virus acting on the CD4 cells, which are present on the T helper cell. As a result, T helper cell number will be drastically fall down. Without T helper cell, B cell never divide into plasma cell and memory cell. Then if there is a no plasma cell, there is a no production of the antibody. Now every student listen. Whenever there is a no production of the antibody, what happens, friends? Our entire immune system is getting compromised completely. Listen to me, listen to me. Entire immune system getting compromised. What do you mean by the word compromised? Even though we get a common cold, even though we get a common cough, we can't tolerate that small symptoms also, because body's immune system is not at all responding. Now that what we can call weaker immune system. So during that weaker immune system, there are certain opportunistic pathogens are there. Friends, don't get confused with my words. What do you mean by the word opportunistic pathogen? Opportunistic pathogens, friends, they cannot attack on our body whenever our immune system is stronger. They can't. If they want to show their action, our immune system should be weaker. So they are waiting for an opportunity. What is the opportunity that they are waiting for? They are waiting for an opportunity where the immune system is completely become weaker. Then they enter in our body and then they will show the carcinogenic or cancer activity. So friends, best example, Kaposi's sarcoma. Kaposi's sarcoma not caused by the HIV virus. HIV virus will cause destruction of the immune system by that the virus, which is a Kaposi's virus. The Kaposi's virus, which is having the ability or capability to cause the carcinogenic activity, and it will attack on the immune system when the immune system is completely weaker. That's why we call them as the opportunistic pathogen. Friends, kindly do not miss this particular slide, which is extremely important. Okay, friends, till this slide, whatever we have studied is the characteristics. and whatever we have studied is the causes. Till here one area. Okay, now you look into the your 2022 paper. So we got 2 plus 4 6 marks from the characteristics itself. They did not come even to the genetics part also. If you miss the question from the characteristic feature, there is only one way of asking a question in the cancer is mutations. You can also call it as genetics of the cancer, which is extremely important for the exam. Very important, friends. Many times the question was repeated from the genetics of the cancer, and it is very easy to remember. So everyone listen to me continuously for the period of 20 minutes without having any kind of a confusion. Just listen to me carefully enough. Friends, we have very clearly came to a conclusion that the cancer is basically caused by the change in the sequence, alteration in the gene sequence. That is what I told you. Friends, how many number of genes are basically identified which are involved in causing the cancer? Friends, listen, there are more than 100 types of critical cancer genes were identified. What, friends? There are more than 100 type of critical cancer genes are identified. Kindly don't get confused with that particular point. My friends, just listen to me carefully. All these 100 type of critical cancer genes are categorized or classified into just two types. What, friends? There are more than 100 critical cancer genes were identified, and all these 100 types were just simply categorized into two types. And the point is very clear. Based on what we categorize into two types, whether, or in a simple way, based on the overactive mutation, based on the underactive mutation. In other words, you can also call it in other words, based on gain of function and based on loss of function. Just based on these two simple criteria, we categorize them into different types. Kindly don't get confused with that point. And what are the examples of that particular two genes? One we called it as, friends, proto-oncogene. Next one we called, friends, tumor suppressor gene. Kindly don't get confused with that. And I want you to remember, friends, the moment you heard the name of a proto-oncogene, one common point has to strike in your mind. Proto-oncogene, because of the gain of function, it is converted into tumor. We can say overactive mutation. Tumor suppressor gene, because of loss of function or underactivity mutation, it is causing the cancer. And the last and final statement, which is the most important statement you have to remember, is proto-oncogene mutations are dominant. You remember this, the diagrams will be very easy to all of you. Focus. Proto-oncogene mutations are dominant mutation. And friends, tumor suppressor gene mutations are, friends, recessive mutation. Trust me, if you remember this, the concept is very easy. Now you look into this. I'll make it simple. Okay. So let's talk about the first proto-oncogene. Then let's take the tumor suppressor gene. Friends, everyone listen. Just think. Do you think, is there any gene called as proto-oncogene present in our body? Do you think, is there any gene called as proto-oncogene present in our body? No. There is no gene such as proto-oncogene in our body. Friends, listen to me. Everyone listen. Any gene which is present in our body which is acting normally. What, friends? Any gene present in our body which is acting normally, undoubtedly we will be calling it as the proto-oncogene only. There is no difference in that. Any gene, if it act normally, proto-oncogene. Every student listen. Now I have told you one word very clearly. Because of the overactivity mutation, because of the gain of function only, the proto-oncogene converted into oncogene. That is the direct 4-marker question asked. Now look into the question, look into the diagram. It is the normal cell, friends. Every student listen, don't get confused with this. It is the normal cell. How many copies of genes are present, friends? You can see it very clearly, friends. Two copies of the genes are present. Yes or no? Of course, yes. Friends, listen to me. Even though two copies of the genes are present, friends, everyone pay attention. Out of the two copies of the gene, friends, only single mutation happened. See here, only one copy of the gene got mutated. My friends, listen. Even though one copy of the gene got mutated, that is sufficient to convert the normal cell into the cancer cell. This is what proto-oncogene. To convert the proto-oncogene into the oncogene, even one mutation in one copy of the gene is sufficient. What will be the reason? I have told you the proto-oncogenes can always shows the dominant mutation, which means one copy of the genes get mutated, that is sufficient. Even though one copy got mutated, whether it is second copy is present or absent, it doesn't matter. That also never works. Don't get confused. If one copy got mutated, friends, this also stops working actually, because the mutation is dominant mutation. Don't get confused with that point. So what I would like to say, the proto-oncogene is getting converted into oncogene via overactive mutation, via gain of function. Unnecessarily it's gained a function which is not required. Unnecessarily it gained a function which is not required, because of which it is converted into oncogene. Kindly don't get confused with that particular point. Next one, friends, underactivity mutation. What do you can also call it the underactivity mutation? Underactivity mutation we can also call it as, friends, loss of function. Kindly don't get confused with that particular point. Now you listen, and I have told you to remember one more important and unique statement. Underactive mutation means what, my friends? A recessive mutation. I have told to all of you. Is it? Yes. It is the recessive mutation. Friends, don't get confused. Now every student just listen to me. When I use a word recessive mutation, you have to understand loss of function. Here, if only one copy of the gene get mutated, the gene never ever lose its function. What, friends? If one copy of the gene got mutated, the gene never ever lose its function. Okay, first of all, listen. What is the best example for this underactivity mutation? Every student pay attention. The best example is a TSG, tumor suppressor gene. Everyone listen my statements very carefully. What you have to understand by the word tumor suppressor gene? We're gonna read it very elaboratively. The name itself indicating na. What is the function, you tell me? The tumor suppressor gene suppressing the formation of a tumor. Correct now, friends? Suppressing the formation of a tumor, that is the function of a tumor suppressor gene. Now listen to me, friends. The tumor suppressor gene having two copies of gene. Is it? Friends, out of two copies of gene, only one copy of gene got mutated, friends. Only one copy of gene got mutated. Do you think this particular normal tumor suppressor gene is going to lose its function? No. Still it works normally. What's the reason? I have told you here it is a recessive. Listen, there is only one copy of the gene got mutated, friends. Then there will be no effect of mutation in one gene copy. It will work like a normal cell only. So ultimate function is what? Blocking the tumor formation. It will block the tumor formation. Just one mutation only occur. But friends, if there is a second mutation occur, what's going to happen? It is going to inactivate second gene copy also. Friends, every student remember, whenever it inactivates the second gene copy, now see here, first gene lost the function, second gene also lost the function. Then how does the tumor suppressor gene is going to protect us from the tumor, in preventing the tumor? It can't. Then what happened, friends? There will be a tumor suppressor gene converted into oncogene, which leads to the cancer. So don't get confused, friends. Proto-oncogene is the dominant and gain of function. Only one copy of the gene got mutated, that is enough to convert into oncogene. Tumor suppressor gene, recessive, loss of function. 2 copies of the gene has to lose its function compulsory. Okay, so now we understand how proto-oncogene converted into oncogene. We also understand how does the tumor suppressor gene also can be converted into oncogene. Now let's try to understand different ways how the proto-oncogenes are getting converted into oncogenes and examples. And we have to also discuss most important one, which is widely used to ask in the exam, is the tumor suppressor genes. We have to read at least 4 to 5 tumor suppressor gene. Among all, the most important one is the P53, which is very widely asked in the exam. After that, P21, then very important one, and in this 2022 paper which we got is PRB protein, correct na? And we should also read about the P16. At the same time, friends, we should also read the breast cancer gene, BRAC1 and BRAC2. These 5 genes we have to read very briefly and very elaboratively in terms of tumor suppressor genes. Now let's start with the first one, overactivity mutation. That is, how does the proto-oncogene is basically converted into the oncogene. Friends, every student please pay attention and understand what exactly I am saying here. We clearly understand proto-oncogene is converting into the oncogene, friends, just because of the dominant mutation. There is no doubt about it. Now the question is here, what are the various chances are there, or what are the various reasons, basically what are the various reasons why a proto-oncogene is converting into the oncogene? There are multiple reasons are there. Listen to me. First, one of the major reason, deletion, or I will give you with examples and I'll explain you elaboratively. Deletion or point mutation in the coding sequence, then gene amplification, and most important one, chromosome rearrangement. So here you can take an example of transversion. We will read in the genetics chapter. You can take an example of translocation. You can take a multiple examples. They can also causes proto-oncogene to convert it into oncogene. So friends, every student look into the picture very carefully here. How does the deletion or a point mutation causing a tumor? Every student listen. When I use a word, friends, deletion or point mutation, please kindly don't conclude that there is a deletion in the many number of nucleotides or many number of codons. Hardly, friends, there is a mutation occurred in deletion of just one base or one nucleotide, that's all. But the point you have to remember, friends, mutation occurred in only one nucleotide, that's okay. But the mutation or deletion occur in the coding sequence. You all people know very well, coding sequence is extremely important to code for the protein. Even though there is out of 100 nucleotides, if 99 nucleotides are normal, if one nucleotide got mutated in a coding sequence, the entire reading frame will be changed. If the entire reading frame will be changed, I am not using a word that protein is not getting synthesized. There is no doubt protein will synthesized, but we can use the word the protein will become either hyperactive or the protein will become hypoactive also. It depends what kind of a protein and what kind of a function it has to be performed. Let me tell you one example for this deletion or the point mutation. Everyone pay attention, look into the diagram very carefully. Look into this, friends. Proto-oncogenic receptor protein. Okay, I have used a word proto-oncogenic receptor. Now you only tell me, when I use a word proto-oncogenic receptor, does it is a cancer receptor? No, it's a normal receptor. I told you it is working as the normal receptor, then undoubtedly you can call it proto-oncogenic receptor. So we have taken two examples of the proto-oncogenic receptor, HER2 receptor and EGF receptor. We all people know very well. In the yesterday class only we studied, day before yesterday class, epidermal growth factor receptor, name itself indicating, right, it will be associated with the many kinds of a growth factor, even HER2 receptors also. They will be responsible for what? Associating with the growth factor. You all people can look into the structure very clearly, which are having an extracellular domain, which are having a transmembrane domain, which are having a cytosolic domain, which you people can understand it very clearly. Now let me tell you, my friends, what's happening here. There is a point mutation occurred. There is a small deletion occur. Where there is a small point mutation or deletion occur, my friends, in the transmembrane domain. Friends, what is transmembrane domain? We know very well. With the help of transmembrane domain only, it can transpan the plasma membrane. Every student listen to me. In the transmembrane domain, what kind of amino acid is present? Originally, friends, valine amino acid is present. Every student listen my statements very carefully. Friends, you know what happened? Because of the point mutation or deletion, the valine is replaced by the glutamine. Now every student listen to me. In day before yesterday class, we have discussed in 2 to 3 classes very briefly and very elaboratively that the receptors are always monomers, which are inactive. Upon ligand binding, what did I tell you? Upon ligand binding, the two inactive monomers come close together. They are going to form the dimer. But friends, you listen to me very carefully. Listen my words. Because of the point mutation, friends, listen. In the absence of ligand only, the two inactive monomeric receptors joined together, and friends, even they undergo the process of autophosphorylation, and it can show the response continuously. Undoubtedly, it can show the tumor, because division, differentiation, development. Now here division is uncontrollable, undoubtedly give rise to the tumor. Such a simple step it is. Friends, here the mutation occurred where? In the transmembrane domain. Now you look into the EGF receptor. Extracellular domain is normal. Transmembrane domain is normal. My friends, you know, right, the cytosolic domain. The cytosolic domain got mutated. What do you mean by the word? What do you mean by what do you mean by cytosolic domain got mutated? Originally, upon ligand binding, cytosolic domains get activated, they become dimerized, they undergo the autophosphorylation. But there is a mutation in the cytosolic domain. As a result, in the whether there is a presence of ligand, absence of ligand, doesn't matter. The cytosolic domains are expressing constitutively, means continuous expression, and they will undergo the autophosphorylation, and they are showing the response continuously, which is leading to what, friends? A tumor. So see here, the line was very clearly written. Cell surface receptor can cause excessive proliferation. Why it is causing excessive proliferation? Either because of the point mutation, or because of the deletion, or because of the constitutive expression. Simple. Okay. So friends, that is about proto-oncogene. So in the tomorrow class, we will complete the tumor suppressor genes completely. Okay. By that, tomorrow I will finish it off. Is it clear, everyone, till now? Please let me know. Let me know. Is it clear for everyone? Let me know if you have any kind of a doubts. Friends, as I said you, the PPT will help you 100%. Okay. Read it thoroughly, and my sincere request, if you find the time, keep the Pathfinder with you and read the PPT. Pathfinder with you and focus on the PPT. By that, whichever the points that are not there in the book are covered in the PPT, which are not there in the PPT covered in the book. I hope it's clear for everyone. The PPT was uploaded on the website. You can check it. Okay friends, I already told you, I am requesting all of you revise molecular biology today and tomorrow. On Saturday we will solve all the PYQs of unit 3. Okay. Okay friends, that's all for now. I'll see you in the tomorrow morning class. Bye bye.